Have you ever wondered how tiny insects navigate this huge world of ours? How do they know where to find food, how to avoid trouble, and even how to get back home? In this episode, Professor Anna Stöckl explains how the tiny but mighty insect brain controls behavior of stunning complexity. Hello and welcome back to In the Swarm, where we dive into the minds behind the hive. The insect edition, because today we have our very special guest, Professor Anna Stöckl.
Thank you so much for having me.
So, how do you get into science?
Well, I'm actually a biologist and a neuroscientist by training, and I started studying biology in Heidelberg in Germany. And actually that connection goes back even before I started studying. So in my school days, I was part of an organization that did extracurricular activities for school pupils in Heidelberg, where we could get into molecular labs and we had workshops on biology.
Oh, that's so nice.
Yeah, and I was really very much on a molecular track when I started studying in Heidelberg, molecular biology. But already there, I loved vision and visual processing and what vision tells us about our interactions with the world. And I actually started to really look into vision also during my bachelor's, more on a developmental aspect, on retinal regeneration, how the retina can recover after injury.
So human vision.
Right, or vertebrate vision. It wasn't fish, but the connection to humans was there.
Right.
But I loved it so much that I then decided I want to study neuroscience. So I did a neuroscience master's in Munich, where we really learned anything from cellular neuroscience, but also more systems and organism neuroscience, and already got a little bit of a connection with insects. And from there on, I decided I really want to try. So I've only worked with vertebrates until then. Actually, I added a few more organisms. So I worked with fish, with amphibians, a little bit with mice. And then I decided, let's try insects. And I did my PhD in Sweden on insect vision. After that, I still wasn't sure I want to do insects. I went back to work with mice and mouse vision in Finland. And then I committed. And I went back to Germany, took the insects with me. And essentially, since then, I've been working on insects. First in Würzburg in Germany, which is kind of in the center. And then I've moved to Konstanz 2 years ago with my group.
Okay. Insect. I think many people have a very, maybe a bit stereotypical, maybe rudimentary image of what an insect eye looks like maybe a fly, right? You have like all of these tiny little black dots that forms like these huge kind of goggle-like pair of eyes. What is it like to see as an insect? You don't really have this idea that actually they would see the world like we do.
Yeah, it's very true because the outside of their eyes looks so different, right? But actually the visual processes that go on are not that different from ours. And so very much like in our eyes or in a camera, the world is, if you want, is rastered into different pixels. And on an insect eye, you see this kind of raster already on the outside in the different facet lenses, all the tiny lenses that make up the compound eye. But we have the same kind of sensory rastering in our retina in the eye, and a camera would have it on their sensors. So we all essentially have different channels and split the image into different pixels. I'd say the big difference optically is that we focus all the light with one big lens, and insects use many tiny lenses to achieve it. And one consequence of that, if we really want to compare humans and insects, or one of the consequences of this is that insect vision is much more coarse. So if you want to imagine, they have much less pixels and much broader pixels. So they see the world in much less detail. They don't see the super fine details that we can see.
So they don't have 4K vision.
No, they definitely don't have 4K vision, but they have other powers. And they see much faster than we see. So if they would watch a movie that we watch, they would see, well, essentially lots of, if you want, individual pictures. So they're essentially flicker fusion rate, the rate at which they see separate images as one, is much, much higher for many insects than for us.
So now a lot of people will just have this aha moment of like, I see a fly on the table, I feel like I'm very fast, I go to slap it, it's gone, it saw you coming like a mile away.
It's exactly that. So the trick to get the fly, and you can actually do that, you could try, is to be super slow, approach it without making any fast movements, super, super smooth, and you could actually capture it in your hand. I've done it.
Is that like an example of dynamic vision?
It's certainly an aspect of the dynamic. So what we mean with dynamic vision and what interests us is how does the visual system change and adjust when, for example, we don't look at a fly or a bee that's flying around during the day, but a moth that's active at night, and there's a lot less substrate for vision. There are a lot less photons around, and the photons that arrive are much more noisy than during the day. How does their vision work? And within that, maybe that's the second big dynamic thing is, even in the same light intensities, if you picture yourself being in the forest and moving out of the forest onto a meadow or a clearing, the light intensity changes. And even worse, if you're a nocturnal moth and you fly around and you come by a street light, the light intensity changes massively. How does their visual system adjust to that? So that's the actual core of the question of dynamic vision.
Because actually, I have two questions of many. One, what do you mean by photons being noisy at night?
Yeah.
And two, I think this is something maybe the listeners already have in mind, but don't we just assume that moths just get very confused, like in the urban setting? Please tell us that they're not.
Let's see. Maybe we start with this and then we get to the noisy photons, because that's also related to how they actually process this information.
Okay.
It certainly impacts nocturnal animals, and we do see moths flying around lights and being attracted by them. The interesting thing is we don't actually know what proportion of the moths that happens to, because we only see the ones that get stuck. We don't know how many of them just flew by and were perfectly fine. But there have been some recent studies, and it's really very interesting that they show that, yes, moths that were flying in a large field, like a kilometer-wide field where they were tracked, were impacted in how they fly by the lights, but only 5% of the whole population was actually trapped. So many of them managed to fly by. And I think that's really interesting, something we are very interested in. How do they do that? What do they do different than the ones that get stuck? And then eventually, how can we essentially help them? How can we organize our light sources and maybe shield our light sources, that we help the natural mechanisms that they already have to maybe not get trapped by the light and fly by, and support that.
Right, so the 5% who actually gets trapped by the artificial lights would be able to actually utilize what they already have and not get trapped.
And I should say this 5% is one single study with 2 species of moths. So this might look very different for other species. This might look very different in other contexts. But it's the first one that really gives us a number and an insight. And at least I think the important thing to take away is maybe not the 5%, but the fact that not every moth that flies by a light gets trapped.
Right.
Many of them might still get impacted because they might spend time passing this that they would otherwise spend on finding mates or finding food sources. But yeah, for us, it's really interesting to understand how are they negotiating such artificial lights, that also we have a very good intuitive understanding of how hard this is when we get blinded immediately and we have to somehow cope with it. How does their visual system do it? And other ways that we can help them with it.
And actually, now that you mentioned blinded, how is it that moths, like, don't they get blinded when they just try to like fly into this big light source?
Yeah, there are different theories, and I should say none of them has been, like, probably none can be definitely proven to be the one reason, because there are probably different reasons for why they get trapped. One theory is definitely that essentially as they get closer to the light, it blinds them or it overwhelms their visual system. It starts to adapt. But then they're at the light and they've adapted, and now that's their basic setting. So flying back to the dark would mean that you don't see anything because your eyes, it's very much similar for us actually when we're in a light room and we go out to the dark. It's pitch black, we don't see anything. That's one theory, that they can kind of not get out again because their system is adapted to the bright light. There are other theories that they might mistake the lights for celestial cues that they normally use to navigate and then accidentally circle around. There's been a beautiful study that recently came out that showed that it's also light sources can mess with a reflex that many insects have, that essentially they use the brightness of the sky to tell them where up is. So in addition, or maybe also alternatively to what we use, which is gravitation sensing essentially, they use the brightest cue in their visual field to know where up is.
The moon.
And well, actually the whole sky, essentially. And insects might think that's up, and if you turn your back to it, you start spiraling. Around it. It might be one more reason of why they get stuck.
Okay, so can you describe what you're doing in the lab right now with the moths?
Yeah.
In the big imaging hangar that we have here at the cluster.
So most of the studies that we've been doing and other labs have been studying movement and flight strategies in very, very constrained spaces.
Right.
Simply because that's the tools we had and that's the easiest to film them in the lab, to track them, to know where they're flying. But the problem is that in these constrained spaces, the animals might never actually show the real behavioral repertoire that they have, because a moth can move kilometers over one night, and flying in a less than 1 meter cube flight cage really has very much little to do with what they actually do in nature.
Right.
And that's where the hangar comes in.
Yay!
Because that's really, I would say, one, if not the only, then one of very, very, very few places in the world where you could track tiny insects on that scale. And that scale is, well, what we can really efficiently track is 10 by 10 meters by 5 meters height. And that's really bigger than anything we could ever do, which is why it's so exciting for us to use it. And I mean, already the first experiments, the first pilots we did were super exciting, because we put our moths in and we saw them flying in like the lowest light conditions we did, which is like starlight, massively faster than we've ever seen them flying before. So we always assumed when the light gets low, the moths fly slower. And they might still, so we're still looking into this and comparing it to bright intensities, but they flew so much faster than anything we have ever seen, than any other studies on these moths or other hawk moths have ever seen.
Just because the
room is big and they can actually really use the whole repertoire of what they can do. Which also makes us rethink of what we were actually thinking, how they shape their visual inputs, because clearly they can absolutely fly fast.
In low light conditions.
And that's interesting, and we can go into this a bit later. It's interesting because we know that actually the visual system gets slower when it gets dimmer. So when the light gets low, they actually see slower, but they still fly super fast, which is very puzzling, very interesting.
That's amazing.
Yeah.
Oh, wow. That's so cool. Okay, but I think we've been talking a lot on one part of your repertoire, which is dynamic vision.
Right.
And now maybe we can touch a bit on pattern vision.
Sure. The interest in pattern vision is actually a very specific one, in that pattern vision seems to be very normal for us humans, because we use patterns all the time, like in our letters that encode information. We use symbols all the time that we use for communication. But it's often considered kind of and I say this in quotation marks, which you can't see here, but like as a higher
We're doing
cognitive,
it quotation marks.
Exactly. As a higher cognitive ability. It's something we very much ascribe to primates and humans. Birds can do it. But insects can recognize patterns. They can learn them and they can memorize them. And even cooler, they can recognize them when they are modified to some degree, not as invariantly as we humans can, but to some degree you can modify the contrast with honeybees and hawk moths that we tested. You can even invert it, so you can have background and foreground inverted. They will still recognize the pattern that they learned. You can change the size to some degree, rotate them to some degree. So they have, in limits, but an ability for invariant pattern recognition, which I find super exciting, because now we can dig into how does this tiny nervous system compare to ours. An insect brain has less neurons than we have in our eye, like in our retina. How does that nervous system have the ability to memorize patterns and to recognize even if we modify them? That's really the question behind all of this. And we are investigating it in a very specific context, which is also the context that our moths and that bees use pattern vision and pattern recognition, which is the interactions with flowers. Yeah. So we're also interested in what do these patterns mean for them on flowers, and how do they actually use them to find nectar and interact with the flower.
Yeah, because actually it is true that a lot of scientists consider like pattern recognition and pattern guided behavior as like something a higher level. That you see in animals considered higher level, but in flowers you get all sorts of patterns. On different insects you get all sorts of patterns. And you mentioned that you're looking at insect models that specifically work with patterns on flowers. How about conspecifics or like,
And actually with the butterflies and moths, we would be in the right place, right?
Right.
That's where you find beautiful patterns on the wings that are likely, and there are some studies, and I must say I'm really not an expert on that side, are used for communication, again, mainly between males and females, but also maybe between males that are territorial. So that's definitely a second aspect of pattern recognition, where very little to nothing is known how this is actually processed in the brains of the insects and how they actually obtain this ability. We know they can recognize patterns also on wings, but how they're doing this, we really know very, very little about.
And actually, this is something that I've always had at the back of my mind, but it's never really fleshed out as a question. Like when I watch, I don't know, documentaries about insects and all of these things, they always talk about like, oh yeah, these insects evolve these patterns to either blend in or either intimidate or become dominant. And I'm like, but they don't know what they look like. Do they know what they look like? Like, and how does it work? Like, it really kind of puzzles me at the back of my mind. But when you're just sitting in front of the television, as like a 10-year-old, you're like, I'll take it. No, and Sir David Attenborough has told me that this is so.
And it's an interesting point, right? Yeah. And I think it's something that I think is really interesting when you work with insects. You get reminded about it all the time. That is, it might also not have to be that complicated. I mean, an insect doesn't need to know what it looks like for its pattern on the wings to work as camouflage. Right. A male that's looking for a certain feature on female butterflies' wings might not necessarily need to essentially recognize the entire complexity of the pattern. Maybe it's just using specific features, and these features are nicely contrasted by the rings and things that are outside it. And the same for flower patterns. Maybe they are using relatively simple features that to our eyes, these patterns look very complex, but they're not actually using the entire complexity.
Yeah.
And that goes for many, many insect abilities. They have amazing abilities, and they're very, very well developed, I would say, in where they have to, like flight control in insects. I keep saying this, like try to build a robot the size of an insect with the control computer the size of an insect.
You can't!
And make them fly like this. You can't.
You can't.
But it doesn't necessarily mean that everything that you see insects do that we might even anthropomorphize a bit is actually super complex.
Exactly.
And I think that's the fascinating thing, where they have found simple solutions to seemingly complex problems. It is, and that's something we're really actively working on. A question, for example, with the pattern recognition, what features are they using? We know that they really like radial features, which makes a lot of sense. If you look at flowers, they're always lines kind of radially converging onto the center.
Into the middle, yeah.
Also, the petals themselves make these forms. And we know they can discriminate them from more concentric features. But what is it actually that they're taking from there? Can we reconstruct essentially the kind of algorithms that underlie this? Do they have filters for these radial features? Do they look at the center versus the surround? What is it actually? How's their pattern recognition essentially composed? I'm assuming, like that's what I would expect, that it is not as complicated as that like the pattern recognition that we have and that primates have, or even that most neural networks can do by now. But that's fascinating because it shows what you can do with such a simple system. And we'll see if it's simple. Because if I've learned one thing from insects, you make a simple hypothesis, you test it, and, well, it's actually a lot more complicated than what you thought it would be.
Yep, I'll take that. I'll take that. That's the thing. I think that's true for most of science. Yeah. That it's like you have a very simple, elegant hypothesis, and then the insect comes in and it's like, no.
Right. Sometimes it works. I've had cases where this worked, and it's almost too good to be true, but yeah.
So we've talked a lot about how the neurons are organized and how they all work in the visual system. I think one of the biggest questions I have in my mind right now is, how is it possible to study neurons and in live animals?
Well, we have different methods to actually record what the neurons are doing. But I would go a step back because we can actually formulate hypotheses of what we would expect, which is always a good starting point before you go in and actually look what the neurons are doing, already based on behavior and based on neuroanatomy. And we can start with the neuroanatomy because that's what we kind of left off with, these neurons that have long processes that pool information from neighboring visual units. These neurons have actually been described more than 100 years ago by Ramon y Cajal, one of the great founders of neuroscience, who described these neurons and already hypothesized that the processes might be integrating information. And over many, many decades after, people have described them in many different insects. One of the beauties of insects as well, you can actually find neurons again. You can recognize that this is the same type of neuron. They have such conserved brains that you can actually do this comparative work down to the single neuron level, essentially.
Oh, wow.
And so that hypothesis was very much driven by the neuroanatomy, that these neurons likely integrate information. And so then we had to do the last step, which is to show that they actually do it in the animals that actually see, and we can check what the neuron's doing while we're showing it stimuli. And so what we have been using a lot, especially to target individual neurons that we also want to check that it is that neuron, is intracellular electrophysiology, where we use a very, very, very fine glass capillary that essentially is finer than the neuron itself, and we can poke it into the neuron. We can also put dye into this glass capillary so that we can record its electrical activity, and we can put the dye in when we're done, and we can check which neuron we recorded from. So that intracellular electrophysiology is really the way to go when you want single neurons and when you want identified single neurons, because you can trace them in animals where we don't, especially in animals where we don't have all the genetic molecular techniques, where like in fruit flies, for example, or what you can also do in mice where you can label neurons in other ways. Yeah, and it's interesting because it's a bit like a fishing expedition, I should say that, because people imagine that we look into the brain and we see the neuron and we poke the electrode in, which is something you can do in some systems, especially in systems where you can label the neurons genetically beforehand with fluorescent dyes. But actually what we have to do is we have to essentially get a window to the brain. We see the surface of the brain. We, over time, learn how it's oriented and angled. And we have to put the capillary in. We don't see any neurons. We just see the coarse surface structure of the brain. And we learn by experience where to go to find these neurons and to find this part of the brain. And then you're either lucky or you're not. And you actually, interestingly, it's mainly auditory. You have to listen. So you get to hear. So we're transferring the electrical activity into, sound essentially through loudspeakers. So we can listen to the buzz of the neurons and the action potentials they fire, if they fire any. And over time, you actually learn to recognize your neurons by their firing patterns and or their baseline activity of how they respond to stimuli. Yeah, and so we go through. We don't see anything. We just step blindly through the brain until we hit the neuron we want. And we can, of course, select which neurons we want because we can show visual stimulation on the screen that the animal sees. And then we can select which neuron it is and we can test it with different stimulation.
I'm trying to contain how mega cool I find this. You're just like, so we just learn how to listen to each and every single neuron in this insect brain over time.
To be fair, I don't think
anyone... To perform surgery, like...
Don't think anyone knows each and every one, but you know one. That's the important thing.
That's the one. That's the one I have to know. It's like, oh gosh.
That's it.
So what you've described so far seems to me, actually is very obvious to me, as a pretty invasive method. And we always talk about ethics when it comes to animal research. I actually don't know much about insect ethics. Can you tell me more about that?
Sure. In insects, you essentially don't need ethical permission to do invasive experiments or any experiments. Where the permissions is needed, and I think that's one important ethical aspect, is for collection as well. Especially in Germany, any intrusion in ecosystems requires permission. So for example, the moths, one of the species of moths we work with, we have colonies of them. We breed them ourselves in the lab, but we regularly need animals to found them again, to like have fresh animals in the colonies. And we can collect them. The animals are not... all the animals we work on, all the insects we work on, they're not endangered, not red listed. So, but we still can go out and collect them. That's a bit of a misconception that if an animal isn't protected and not endangered, you can just go and collect it. You can't.
No.
Not insects either. So for that, we need permission. But for the specific work, we don't need to apply for permission to do intracellular recordings or neuroanatomy that we take the brains of the animals. That doesn't mean that we don't consider what we're doing. So we very much make sure that we only use as many animals as we absolutely need for the studies that we do. That's, I think, one important consideration. And there is a bigger and bigger discussion about the ethics of insect experimentation, or generally insect ethics, because it hasn't really been an issue. And it's getting more relevant. The discussions are interesting and I would say controversial, and I don't really have an answer to it either. I think this is something that also research will show in the future as to the level that insects respond to invasive methods the same way that we do.
Right.
And I mean, the big question when you do invasive experiments is to what pain and stress.
Right.
And there's certainly no questions that insects have responses to being grabbed, to being attacked by predators. But to what degree these responses are connected to the feeling of pain, the experience of pain, is not clear at all. And that's where it becomes interesting, because then you have to dig into what actually does it mean to feel pain, to experience pain. What level of cognitive abilities, of conscious abilities do you actually need for this?
Yeah, no, for sure. I think you can never escape really experiencing the world and considering different things in your world as a human. But I think it's also really important to kind of, keep that open question in mind. Like, okay, this is very human-centric.
Right.
That's fine. I am a human. But also, what are these human values to an insect or to a colony of insects?
Well, and the other thing is, I think, do research that is relevant and that has an important objective. And I think that's also a difficult thing, right? Because what we are doing is very much basic research. And I was always driven by this really impulse of understanding how things work, understanding how the world around us works. But that doesn't have an immediate application. And you might be more prone to say, well, research is only relevant if you can do something with it. And it's only relevant to use animals for this research if it has an outcome. And it's an interesting argument, because I do not agree. I think that's one important result of research. With understanding the world better, with understanding insects better, how they work in dim light, how they work with, like, how they're impacted by artificial lights, how they interact with flowers. We also understand better how we can support them, how we can actually support conservation. And I think thinking about that and thinking about the impact of your research is really important when you have to justify also to yourself that you're using animals for this research.
No, for sure. I think knowing something or not knowing something, or realizing that you don't know a lot about all the things that go around, that happens around you in the world, really gives you a different perspective. And I kind of want to get back from the insects back to you again as a researcher. What excites you as a researcher?
Part of it is the questions, and we talked a lot about why I'm so fascinated by vision and insects, but I also really love the process. I mean, it's a job where you can ask a question driven by curiosity, of course, on the backdrop and the shoulders of research that has already been done. But you can ask a question, you can formulate hypotheses, you can come up with a way to test it, get the methods together, build your own setups, actually investigate it, analyze the data, and have an answer to this question, which you can essentially create this entire process from scratch and get to the product yourself and do something that no one has ever done before. And it's an amazing process if you think about it. And it's really what makes me super excited. And it changes a bit as you run a group, as I do now. I'm not doing so much of the practical work anymore, but it's still the same process as we can think about coming up with experiments, designing them. And what I really enjoy about it now is actually seeing how people do this, how different people do this, how they approach it, and me being able to guide it to some degree, and really actually going beyond what I could have done and seeing all of these different ideas of how to do it and embracing them and actually doing a lot more than I ever thought we could achieve with a group. So I love this process, and all of the more tedious things that come with it to make this process possible are really worth it, I think, for experiencing it.
That's only good. Anyway, thank you so much for your time today. I'm super stoked, and this has been In the Swarm. Stay tuned for next time. Bye!
Bye!